Great rewards don't wait, so why should you? Through the Invigor Book and Buy Grower program, you could earn a $30 per bag rebate when you book all of your Invigor hybrid canola by November 14th. Talk to your retailer. Terms and conditions apply.
Join the next wave of growers switching to the next generation of phosphorus fertilizers by visiting Phospholutions.com. Hi, everyone. Welcome to the Agronomists. I am your host, Lyndsey Smith, and I am, as always, super excited to bring you tonight's broadcast. We're going to unpack some really cool technology, and I can't wait to dig in. But of course, before we do that, a few housekeeping items. Let us know, of course, in the comments where you're from and what the weather is like, just like Jeanette has done, where I hate to say it for those of us here in Ontario suffering from drought, but Manitoba is hogging all the rain. And it's almost harvest, you guys, or harvest has started. Give it to us. Usually we want to send it that way. Anyway, it's been definitely a trying season, and that will be one of the topics for tonight is what kind of things can the technology we're going to talk about actually manage. Before we get there, though, remember you can collect CU credits for watching The Agronomists. Head on over to realagriculture.com/agronomist. Let us know you took in the show. We'll get you those CU credits. And while you're there, sign up for the newsletter that reminds you what's coming up and reminds you to go and collect those CU credits. So there you go. Okay, now let's talk RNAi and CRISPR technology. What on earth are these things? To have that conversation, let's bring in tonight's guests. We have Chris Manchur with the Canola Council of Canada, based at Winnipeg, and Dr. Liping Wang with the University of Guelph here in Ontario. Welcome, Chris. Welcome, Liping.
Good evening.
Good evening.
All right. I am excited to talk about RNAi and CRISPR, two technologies that are maybe similar in some ways, but very different in lots of others. But I think we probably group them together for good reason. Tonight, we're going to unpack what these technologies might be able to solve for us. But before we jump into that, Chris, I'll start with you. Now, agronomy specialist with the Canola Council, but this topic is near and dear to your heart. So tell us why.
Yeah, even before I was even thinking about being an agronomy specialist, I thought I was going to be strictly in research for this. And during my master's, I was working at the U of M, and during COVID, looking at stuff like this, which was RNA interference. Now, a lot of my work was on optimizing protocols and such, but I was working on some pathogens and amongst a lot of other people that were looking at stuff that we're probably going to be talking about today. So it was pretty interesting.
Mm-hmm. All right. And Liping, tell us about the work that you do at the University of Guelph, both on this topic and, of course, what you've done before and what you're working on now.
Sure. My research background is carbohydrate metabolism, including lipid metabolism and starch lipid metabolism. So recent years, when I joined in University of Guelph, I have done amazing work. So we try to manipulate starch metabolism in canola, and we have achieved a high yield under greenhouse conditions. So right now we are doing the field trial. So the field trial is ongoing. Probably I will talk about more details later on.
Okay, exciting stuff. This is super cool. Okay, so what are we actually talking about? And that's really the crux of where I want to start this conversation. For those listening, I think in the past few years, we hear the term CRISPR, Cas9, we hear RNAi maybe a little bit more. I know even within Real Agriculture, we do have a few clips that we'll play over the broadcast tonight, sort of touching on some of the things that are, really showing some great promise on what we can do. But I think it'd be really cool first to sort of actually talk about, what is RNAi technology? What is CRISPR technology? And so, Chris, maybe I'll go to your slides first, and then Liping will go to yours because I think you've got some great ones that show how CRISPR sort of works. So, Producer Jay, if you would bring up Chris's slide 1, we'll start there. I'm going to start with Chris. I stands for interference. What the heck? Please explain.
Okay, so RNA interference can get pretty crazy because every organism has it, but luckily, because every organism has it, we can kind of hijack, these internal processes, right? And so when we're thinking about RNA interference, we're thinking about targeting pests, just like we do with pesticides. Now, pesticides have been around for quite a long time, and after quite a bit of study, we see that these chemicals will target a certain enzyme or a protein, and once that protein or enzyme is no longer functional or occupied with that chemical, it's a loss of function, and then it causes that effect, which we like to think that it'll kill that organism in most cases, or at least suppress it. RNA interference, all it does is we're going one step backwards in this whole process. We call this the central dogma. Maybe we've heard of this, back in high school, where we go DNA, RNA, protein. And so RNA interference, what we use is this molecule, which is double-stranded RNA. Usually we think of it as single-stranded. And then we spray this, or we put this into a plant through, genetically modifying it. And once this actually gets uptaken by that target organism, say like a pest, a fungus, an insect, maybe even a weed, it'll chop it up with their own machinery, because every organism pretty much has this. It'll remove all that RNA. And if you don't have it in the first place, you can't have the protein. And then it causes that same effect as a pesticide.
Okay. I'll be honest, I'm still going to need more clarity, but that's a good start, Chris. It's a good start. But yeah, we have to wrap our heads around sort of, and I like this in that if we understand, or if we understand how a pesticide works, then we, as you pointed out, we're sort of taking it a step backwards in the process. And still sort of achieving the same result, but just in a bit of a different pathway, which is pretty cool. So pause for a moment. Mark has asked a very important question, which is how much is sweet corn on roadside stands in your area? Everybody in the comments, go add that, because I, for one, was incredibly blown away by how expensive sweet corn is right now. Okay, corn is one of the ones that we may see some of this, or are seeing some of this RNAi technology make its way into hybrids. So we will be talking about that as well. But Liping, I'll go to you and your slide 2, producer Jay, so to introduce both of the technologies we're going to talk about. CRISPR. Now, I feel like I've heard about CRISPR a lot longer, as far as sort of in the mainstream news, etc. And when I first heard about it, I felt like, aha, we have solved all of our problems. But I feel like maybe we're not there yet. So Liping, what is CRISPR? How does it work?
Yes. CRISPR is kind of familiar for biology people. So CRISPR is short for clustered regularly interspaced short palindromic repeats. So this CRISPR sequence was originally identified in bacteria E. coli in late 1980s. So at that time, people found that as part of an adaptive immune system. So the gene editing breakthrough, this technology came in 2012 when California Jennifer Doudna and Emmanuel Charpentier, they showed that CRISPR sequence and Cas9 protein can be programmed with a guide RNA, and then they start to cut the DNA, genomic DNA, to produce double-stranded break. And then in 2013, several groups, including Zhang Feng at MIT, they developed a technology which is genome editing in mammalian cells. And very soon after, people established this system in plant cells, making this technology a very powerful biotechnology tool. Since 2013, you can see the published paper using this technology to improve crop trait has been surged a lot. So this CRISPR system consistent two components. One is sgRNA, as showed in this slide. Another one is Cas9. So sgRNA, it contains a 20 base pair sequence with a with a sequence of NGG if for Cas9. So this this guidance RNA can guide Cas9 protein to the target DNA sequence, and then this Cas9 protein started to produce double-strand break. After producing a break, the cell will use its own mechanism to fix this break. So usually there are two mechanisms. One is non-homologous end joining. So this will produce random repair. So it can be
Okay.
insertion, deletion, and another DNA repair system is homology directed repair. So this will be very precise. So you can insert it a precise fragment using this break. So this is a very amazing system. It can be very precise and very efficient.
So that is what, and I completely agree, Liping, is that one of the things I think that I'm fascinated by is how precise that this can be, and how, and we've got the applications there, and that's what we're going to sort of unpack tonight, is what challenges to crop production these different technologies can address, and why, as we understand the technology, why some things are better suited for others. What I find like really fascinating is that you can remove, as you say there, undesirable genes. So you can actually sort of clip things out and sort of fix problems, which I think is fascinating. Also, can I just say I very much appreciate CRISPR as a acronym, because I would never remember that whole sentence of what CRISPR actually stands for. So that is pretty fantastic. Okay, so Peter's got some good questions, and these are ones that I think sort of get to some of what we're going to talk about tonight. And that is about how we apply these different technologies to outcomes, right? So what can we actually do with these? So that's maybe, Chris, you've got some good slides on this as well. So maybe we'll go to slide 2 of Chris's, and we'll talk a bit about what these different technologies sort of apply to better or worse, etc. So first, maybe, Chris, your opinion on, as we look at RNAi being applied, where so far anyway has it really been shining and really doing well?
Okay, I think some of the places where RNAi has really been shining, and I'm going to even kind of lump that into with some CRISPR gene editing, is that even before we think about seeing something out in the field, it's really useful for proving studies, proving gene functions in the lab. You know, when we're taking a look at, okay, how do some of these processes work? Because then we can use that for, you know, future studies, breeding, developing even different chemicals. But where I find that RNAi really shines is in these foliar applications and options where we can kind of combine or have substitutions for certain chemicals. And so on this slide, what we're looking at right now, I want to give kind of two different perspectives. One on a commercial product that's, you know, just about being available in the United States, and another one that we've seen that's been in research for a while that actually came from the lab that I was researching in. So, and also, it's really nice to have some visuals, you know, when you have a big bar versus a little bar, and you can really show those comparisons. So there's a company called Greenlight Biosciences who's produced a RNAi mode of action. There is actually now a new mode of action category for RNAi. And they wanted to compare that against, you know, the industry standard, so a regular insecticide. And we see that it's not providing equal protection, but it's pretty darn close, especially when you compare it to that untreated check. So in cases where, you know, we have a really immense threat, or, you know, if there's maybe regulatory pressure on some chemicals, this is where it really shines, because it gives us a new mode of action. It's very environmentally friendly. And usually, and this is usually where we have to see the business case for it, is that it has to be comparable to what we have today. So on the right side, we see it's something similar for sclerotinia, right? We have 4 different leaves here. We have one that's the control, so where they put a little bit of sclerotinia on that leaf, and it just looks really ugly. It's infected right throughout. And this doesn't take too long after. It says 2 days post-inoculation. We know Sclerotinia is really aggressive, but if we can nip it in the bud early, just like we do with fungicides as a preventative measure, then that's going to really help. And so we see the other 3 leaves, there's different gene targets that we're looking at for RNA interference, and they all have, pretty good protection. But we know that there's going to be at least one that's really, really effective, or maybe want to combine some of these chemicals together to provide additive effect. But also the other thing too is look at how much we're actually applying of this dsRNA, 200 nanograms. There's a lot of zeros in front of that decimal point, when we're thinking about grams for that. So it doesn't take much. Just similar to like on the left side, 3.76 grams per acre, right? That's less than, say, some of those common insecticides we see today too.
Mm-hmm. So, right. So we've brought up a couple different aspects to this, right? Is that, does the product work? How does it work? How do we apply it? And or is it potentially in a plant already? And Jeanette asked a great question. And Liping, actually, I want to go to your slide 3. And Jay, so this is on, because you mentioned in your slide, in the first slide, about CRISPR, that improving nutrition potentially of eventual crops could be a use of this. And so I want to talk about that a bit, but Jeanette's question, and so I think that this sort of dovetails into that, is does public perception of CRISPR differ from other genetic modification? So, and that question is somewhat complicated, but I do think, we're not, and Liping, correct me if I'm wrong, we're not adding any DNA here, right? We're simply taking the DNA code that the plant already has, and we're simply modifying it, but modifying with what's already there, right? We're clipping out something that we don't like or that's wrong. So how is it maybe different than our traditional genetic modification techniques? How is CRISPR different?
Yes, you are right. So when the plants finish gene editing, the Cas9 protein and the selection markers such as kanamycin, BASTA, other selection marker, so this protein, these sequences will be segregated out. So in the final homozygous plant, so you will not identify any residue DNA from Cas9 from selection marker. So this is not achievable from the traditional transgenic approach.
And so do you feel, or have you, like, what has your experience been with, because we certainly get a lot of public perception pushback from transgenic or GMOs, or, does CRISPR, does it seem more accepted, or do people still, like, where does it fall in sort of the acceptance aspect?
Well, yes. So I think different country, they have a different policy so far. At least in Canada, I know Canada has a very open mind for this CRISPR technology. At least from our project, ongoing project, we have generated several CRISPR mutant. So this mutant line, canola lines, has been waived from regulation. So that means CRISPR is not considered as traditional transgenic line. So the
Right.
perception, if people know there is no residue foreign DNA being left, so I think the public perception will be different from the traditional transgenic plants. Of course, maybe some countries they still treat traditional transgenic and CRISPR transgenic line as the same, but
The same. Right.
yeah, it is a trend.
Okay. Chris, your thoughts on that? I mean, if we
Yeah,
needed a poster child for anti-GMO, it would be probably canola. When it gets pointed out, unfortunately. So I would imagine you have some thoughts, but your thoughts on the acceptance from the CRISPR side, and then we can unpack the RNAi side, but from the CRISPR side anyway.
Yeah, what I really like to see, and what we've seen also right now, is that we do have a very robust kind of regulatory system that is approaching a lot of this work in terms of that outcome based. What is the end product, and not necessarily on the process, right? Because that the end product is what we're consuming, and what's maybe impacting our environment. And so, as Liping was mentioning, it's we've seen kind of that equivalency for that, which is excellent to see. And I think it does provide a really predictable kind of environment for companies, breeding organizations, life science companies, to actually invest and get these products to market, which is really promising.
Mm-hmm. Now, okay, we've got some great questions about specific applications and where we may run into some trouble, or what's possible and what's not. And I want to get to those. But producer Jay, let's go to our first clip of the night. And this is Haley Catton and John Laurie talking about using RNAi to combat wireworm. So let's go to that read, and that clip, and then we'll go to our first read of the night.
So right now we're just developing the early part of the technology. So it's an RNA technology. So we needed to know what RNA is expressed within the cells of our target species. And so we've collected last summer. Haley and her group collected the larvae and beetles from our target species, and then in my lab we extracted RNA, sent that out to a facility to sequence it. And that came back, and now we've assembled the sequences and have a list of all the expressed genes. And so from published literature and other groups that do RNAi, we have some favorite genes that we're looking to target right away.
So talk a bit about how you kind of determined which species and genes you were going to target.
Sure, I can take that one. So there's been a 15-year survey of wireworms on the prairies that just got published last year, and about more than 90 to 95% of them were of 3 species. So I have here a wireworm field guide for the prairies that was produced by myself and some colleagues, and we're focusing on 3 species here that were like very, very abundant. So this one, and this one, and they all have long Latin names, so I won't list them here. But that's how we determine which ones to focus on, the most abundant 3. And then if we find some success, we can adapt to other species as needed. Large amount of wireworms, but the pest species are only like 5 or 6 species, at least on the prairies. There are hundreds and hundreds of species of click beetles and wireworms in Canada, maybe thousands. Right? It just so happens that 5 or 6 of those species on the prairies just happen to really like our cropping conditions and have become a pest.
Our sponsors for the Agronomists are Rhizosorb, the Farm Safety Roundup, and the Canola School. From preceding seeder setup and checks to pest ID and advice on nutrient management decisions to tips on determining swath timing, Real Ag's Canola School covers every facet of the canola growing season. The Canola School is made possible through sponsorship by BASF Canada and Invigor Hybrid Canola. Learn more at canolaschool.com. Okay, so once again, this is where I need you guys to put in my brain the facts of how this all works. So in that clip, we see where we've identified a pest, we've identified the most common species, and that's an important part of developing this RNAi tool to then target that pest. So that then, Chris, I'll go to you first. Jason Vogt out of Manitoba, maybe you've met. I'm wondering how close we are, I would assume so. I'm wondering how close we are to RNAi application to weeds, like water hemp, where we basically sterilize male or female plants so the natural population can't cross and survive. I love where Jason's going with this. What say you, Chris? Is that something where we could see this go, or what roadblocks do you see?
Okay. I think this is probably one of the best cases where we have to think about gene editing and knockouts versus RNAi. And so it's another way I think about gene regulation, downregulation mainly, versus like completely eliminating that gene. And when we think about, say, in Jason's comment about sterilization, you want to make sure that gene is knocked out and it's not functioning.
Right.
Even when we're thinking about, say, transgenics, which is that GMO option of putting the RNA mechanism or those sequences into a plant, like we may want to do with water hemp, it may downregulate. We'll probably see some impacts, maybe enough to distort that reproductive pathway. But when we have other options, say like gene editing, that may be a little bit better. Now, probably what's going to be the big asterisk to all of this when we're, genetically modifying, natural or wild populations, that comes with a lot of concerns. How is this going to be stewarded? Making sure that there's not going to be any releases of other genetic material, all those other concerns. So in terms of where's the regulatory pathway could go for that, it's going to be a little bit more uphill compared to ones where we're maybe more controlled and more specific. I like to think of that as kind of like, seed treatments for seeds, are targeted to insects that are feeding on it, whereas a foliar insecticide, a little bit more general.
Okay. Liping, do you want to weigh in on that one? Or I want to sort of bring this in more to perhaps on the where your work is on the starch and protein side, because
Sure.
Peter wants to know, and of course he brings it to wheat because that's Peter's favorite crop, which tip of the hat, that's fine. Can RNAi be developed to apply to a wheat crop that would increase protein in a crop like wheat? Is that a possibility? So like we saw on that last slide where there was that discussion about can you change a starch profile or a fat profile, these sorts of things. Is RNAi that tool, Liping, or would that be a CRISPR application? Or how do we sort of find an answer to this if it's possible?
Can I get the question again, Peter's question?
Sure.
RNAi can apply. Oh, okay,
Yeah.
I saw that.
Yeah, there you
Increase.
go. Yep.
Let me think about.
So could we increase protein with RNAi?
First of all, RNAi is knocked down, is not knock out. So it can be increased protein. It depends what you are targeting, what RNA you are targeting. If targeting a negative regulator of that protein, of course the protein can be increased through RNAi.
Okay. So so it is possible, but it does get, to me, it sounds like it gets complicated because it does get very selective. Okay. So now Peter wants to know, so can CRISPR technology be used to insert genes, or would you just use a different transgenic pathway to do that? Or is CRISPR really about just working within the DNA of the plant that you're working with, or the disease or the pest? But is CRISPR really just about working within the DNA, or can that type of technology also be used to introduce genes?
Yes, CRISPR is only to modify genome sequence, which is a DNA sequence.
Yeah. Okay, that's good to know. Okay, so we saw then, Chris, that discussion of wireworm, of first figuring out what we're going to target, which I think it is in a lot of ways very elegant. But like Jason's question, we have some very nasty pests out there, and we probably all have a wish list that we wish like science would prioritize these ones, or please could you fix these ones first? Which in thinking about the applications of RNAi, which pests and or problems do you think we will see solutions for first?
I think the first things is going to be our pests and pathogens, probably with the RNAi. And probably the reason why I say that is because when we're thinking about using RNAi, we can use this as exogenous. I believe that Peter kind of mentioned it as. I like to just call it foliar. We're going to be spraying it just like a regular pesticide. We don't have to worry about a lot of that regulatory pathway or, the breeding cycle, say for canola. We can synthesize the product and spray it on in a timely manner. So, and especially when we know a lot of these insects or pests or pathogens, we know when the timing is, and we can kind of target that to those scenarios. So at least here in canola, we've kind of definitely had a laundry list of different pests, pathogens, emerging threats that we want to kind of tackle. But really, chiefly, it's going to be flea beetles and a few of our major pathogens, which is Sclerotinia, blackleg, and also new on the chopping block is Verticillium stripe.
Yeah. Could you get a handle on that? It's really bad.
Yeah, well, if we can jump over to slide 4, we can show some work that's happening out of Agriculture Agri-Food Canada in Saskatoon. So
like
Dr.
it.
Hussein
That seems
Borhan's been,
hopeful.
it looks great.
Okay.
Not so much that middle one, which is the susceptible to verticillium. They do this with a root dip method inoculation. On the left side is some resistant germplasm that they're making comparisons against. And then on the right side, they've been applying this double-stranded RNA treatment, about a day before they inoculated with verticillium, which is fairly early, but also that's the critical stage. And I mean, from these pictures, I kind of like slide C on the right side a little bit more, right? And also, now we don't have to worry about, maybe there's a yield drag on trying to get those genetics in, or maybe we want to apply this spray maybe only in Manitoba where we know it's a major issue. So we have a little bit of flexibility there. And here's kind of the cool part is that Dr. Borhan's work with this dsRNA, this is a longer sequence, it actually works against blackleg too. So if we go back to, slide 3, we can see that exactly the same product, this dsRNA, and look at the difference here. If we look at slide figures A, C, and E, at different stages, it's not looking too good when it's infected with blackleg. But at those same stages treated, same kind of cultivar, it's Westar at B, D, and G. It looks pretty healthy. I would say there's going to be pretty minimal yield loss for that. So that's pretty promising, I think. And if we can tackle a few pathogens with one product, especially if they got to be applied at the same time, I'm going to like that.
So, I like that as well. Also, I love that Westar is what we're testing these things on. Blast from the past, Westar, everybody. And Jason points out Verticillium stripe is really showing up bad right now. Lots of canola lodging and going flat because of it, and definitely a yield penalty. All this rain on lodged canola, not going to help either. So what I find fascinating about this, and so this, Peter, is one of your answers for we have... and I do have a clip with the RNAi for corn rootworm, and that's a bit of a different story, but also with this. So this is an application very early, which it has to be early for verticillium and for blackleg, but because it's got these, it's targeting 8 different genes, it is actually, from what I can tell anyway, it looks like it's getting really good control of both of them, but it means spraying this particular product very, very early. Like, how early was this sprayed? Like, are we talking like cotyledon stage, or how early are we going in?
It's probably going to be about that cotyledon to 2-leaf stage, which, in all honesty, that's about the staging you want to look at for a regular blackleg foliar fungicide anyways.
That's right, yeah. But how common is that? Like, I know that there are farmers, obviously, that do apply then, but is that, or do you think that because of verticillium, we'll be doing it more?
If we have an effective treatment, I could see this being more commonly done. I think if it's going to really boil down to that ROI, if we can save, 10 bushels an acre, if we have to do that extra pass earlier on, I could see people jumping for that.
Okay. In that example, Pete, good point. What happens if you're late? So that happens. It is a very early infection that occurs. Or do we not have that research yet? But what happens if we miss this critical early window for an application? Can we still make use of some of this technology?
Yeah, I'm going to back up probably just a little bit rather than this particular case. And that's because RNA interference is going to have a little bit of a lag component compared to, say, a pesticide. We saw that it happens earlier in the process compared to targeting the protein. So it's kind of got to work inside the organism a little bit. So trying to figure out, what are the genes controlling it to be quicker, and figuring out that timing and the dosage rate, that's all going to be very important. And that's essentially one of those last steps for optimizing this. Now, What's nice is that you could probably reduce the infection, but we know a lot of these diseases are pretty aggressive, and there's probably going to be a critical amount of, infection where you're applying a little bit of this. It's just the balance isn't there.
Okay. Okay, so Liping, I want to go back to talk about the nutritional side of what we can potentially do. So, Jay, if you go back to slide 3, because there's another question. We didn't get a chance to sort of walk through this. So, explain to me how CRISPR can improve the nutritional profile potentially of a particular crop. And then Mark's got a question about how that may translate to when we feed these crops. Will that potentially change the ROI on potentially feeding some of these crops if we can change the makeup of what they are? So explain to me how CRISPR can be used to improve food nutrition in different crops.
Sure. These are only 2 examples to show how CRISPR improves food nutrition. So on the left side, it's increase the resistant starch. So a starch branching enzyme, this enzyme is very important to produce amylopectin. I need to talk about some background. So starch has 2 different polymers. One is amylopectin with a lot of branches. Another polymer is amylose, so it's kind of a linear glucose. So without starch branching enzyme, amylopectin will not be formed. Instead, amylose will be accumulated. So in a few crops, including potato, rice, and wheat, if you knock out starch branching enzyme, the amylose content will be increased. So usually amylose content is correlated with the resistant starch content. So resistant starch is very healthy for the bowel systems. So in the modern nutrition, so people prefer this higher resistant starch. On the right side, this is an example to show to increase oleic acid content. Oleic acid is considered as a stable oxidation resistant fatty acid. So fatty acid desaturase 2, this enzyme is responsible for converting oleic acid to linoleic acid, which is 18,2, contains 18 carbon and 2 double bonds. So this right picture shows in the soybean, if they knock out fatty acid desaturase 2, you can see the chart, the oleic acid, which I labeled with red. The oleic acid content increased to more than 80%. Actually, in the control line is only 20 something, 20 to 30%. So that's a significant increase. Actually, this oil composition also can be achieved by RNAi because this enzyme has a dosage effect. So even you cannot completely knock out, so it still can increase oleic acid accumulation.
So in looking at these applications of these, the starch branching, so this is the resistant starch, that's better for our blood sugar, right? Is that where that usually in human health, that's like we want more of that for that side. So Mark's question is more on because I think about, obviously we have human uses for many of these crops, but we have livestock uses for them as well. Are there applications where, an altered starch composition would also potentially be more efficient for livestock to do either of you have any experience on the livestock side? Because that also would be fascinating, I think. Or no? We will have to wait. So an altered starch composition would other things then. Jeanette wants to know, how's the upscaling going on some RNAi technology? Will this be commercially available in say 5 years. So we do have sort of limited applications, but Chris, what say you? How do you feel about the sort of commercial availability of some of this tech?
Yeah, I think wonder if Jeanette's probably thinking about synthesizing all of this RNAi, because we can get into transgenics too. We talked about the corn rootworm, that can work.
Yep.
But what's really attractive is when we can synthesize it and spray it. Now that is probably going to be one of the biggest stop gaps for RNAi to become commercial. However, we've come and made significant strides in the past. I would even call it 5 years now. It cost me thousands of dollars to produce a small amount for my lab studies. And those little kits, we're not going to use them for agriculture on large scale. Nowadays, there's been a few different methods. Some are proprietary that say Greenlight Biosciences is doing for their commercial product. And there's even another Canadian company called Renaissance Biosciences who is using yeast as kind of these mini reactors to generate all this dsRNA inside them, and then you break apart the yeast, and then you have a whole bunch of this available. So there's going to be a few approaches for that, but we still have to optimize all this synthesis and making sure that the cost is going to be below what it needs to be to get the appropriate dose of the lab and out of the benchtop into the field now. So that's really promising.
Mm-hmm. Okay. Actually, and Pete mentions it that we do have some of this commercialized already. So we do have a clip on that. So producer Jay, if you would, we'll go to our last clip of the night. And then I want to highlight some of Liping's work as well on moving some of that into commercialization as well, and what the promise of that may be. So producer Jay, if you would, we'll go to our last clip with I want to highlight some of Liping's work as well on moving some of that into commercialization as well, and what the promise of that may be. So producer Jay, if you would, we'll go to our last clip with Adam Pfeffer, who apparently
with the messenger RNA within that corn rootworm larva. It's a very high dose kill, so it's very effective at killing the larva, and it's very specific to corn rootworm. So again, a new novel mode of action, a little bit different method than a traditional BT trait, where it really locks up the guts of the corn rootworm larva and has a very effective kill. And yeah, we've launched it last year with SmartStax Pro. SmartStax Pro has 3 below ground proteins. VT4 Pro has 2 below ground modes of action and the 3 above. So it's really a pretty comprehensive package for both above and below ground insect protection.
So Adam, let's talk corn rootworm. What type of impact is that pest making here in Ontario and across eastern Canada? What have you observed?
Yeah, certainly the last few years we've had challenges with corn rootworm, where we see growers, farmers in that long-term continuous corn situation. And we have had some PIs the last few years with traits like SmartStax in the marketplace. And really, corn rootworm has had a long history in the U.S. of significant challenges. There's millions of acres down there that are really in areas where they really love to grow corn on loam soils. So it's no surprise that it has developed here in Ontario, and is certainly a challenge that we do see continuing throughout eastern Canada over the next number of years.
The Agronomist is brought to you by the Canola School, Rhizosorb, and the Farm Safety Roundup. Keeping safety front of mind is all about communication. With agriculture expertise to help keep farmers, their families, and workers safe, Farm Safety Roundup is a podcast series focused on important conversations with the team of ag specialists at Workplace Safety and Prevention Services. Visit realagriculture.com or find the Farm Safety Roundup on YouTube. All right, so we have a few things I want to cover in this last segment. I want to talk about one of the things you pointed out, Liping, is the potential to boost yield, really, which is when it comes to crop production, one of the major things that we, of course, are always looking for. And sometimes that yield potential is the driver, and sometimes we just have to protect the potential that's there. So these technologies sort of fit into both those baskets. Let's go, producer Jay, if you would, to Liping slide 5. This is your work, your research that you have done, and the ongoing project. Walk us through, tell us what we're looking at and what your work has been doing on this topic.
Yes. This project builds upon our previous findings in the model plant Arabidopsis, where replacement of endogenous starch branching enzyme, again, starch branching enzyme, with a maize starch branching enzyme different isoform. So this transgenic Arabidopsis transgenic plants has shown much larger size and a 300% increase in the total seed yield. So this is in the model plant Arabidopsis. So we are thinking about if we transfer to canola, because both of them belong to Brassicaceae family and they are oil seeds. Even in the field, there will be 1/10 of a yield gain. That will be a great. So Arabidopsis and canola have. So Arabidopsis and canola have a very highly conserved sequence function. So that's why we transfer this approach, this technology from the model plant to real crop canola. We are using CRISPR-Cas9 and then introduce maize starch branching enzyme isoform. So the picture on the left side shows under the greenhouse conditions, you can see wild type and the two representative transgenic lines. So these transgenic lines have increase the total seed yield about 30%. And this yield gain is primarily due to increased number of stems and pods. Additionally, knockout starch branching enzyme in canola results in thicker stem. So this trait of the thicker stem is maintained in these transgenic plants. So thicker stem potentially will offer increased lodging resistance or other benefits. So this will be tested in the field. So this, like Lyndsey, what you said, is boost yield, and in the meantime, it protect because of increased lodging resistance, or probably increase the shattering resistance. So this trace can protect the yield loss. Yeah.
So Chris, Oh, sorry. Go ahead, Liping.
Go ahead. So this phenotype was observed under greenhouse condition, and this year we started field trial. So hopefully, we are looking forward to the field results.
So, Chris, when you see something like this, like super exciting, and then like my wheels get turning about, how quickly can, say, a plant breeder or the plant breeders start to work, picking and choosing some of the other traits that we want, and turn these into commercialized hybrids, right? So that's where my brain kind of goes. And you mentioned earlier sort of that potential for maybe a faster process of working some of this into existing lines, so we don't have that yield drag or that slower introduction of hybrids. So how do you sort of noodle through that of putting some of these technologies together to end up with some really resilient hybrid varieties?
My first thought when I'm thinking about looking at gene editing is that we kind of get to skip forward a little bit in this process, right? Especially when breeding, where when you're trying to get a particular trait across, you have to bring a little bit of other stuff with it. And so if we can use gene editing to kind of select perfectly what are the specific sequences, the functions, replacing with other ones like we're talking about, this is taking a similar genetic sequence from corn and putting it to canola. That makes things a little bit more streamlined. Now, the other thing too, when we're thinking about commercialization, we'll have to see this all in those field trials too, and like, what are those extra results? There's got to be that investment to kind of bring it towards commercialization. And I'm really hopeful that we'll be able to see some of these new technologies provide that, stronger incentive rather than having to maybe have a little bit longer breeding program that is maybe more costly. So there's going to be a lot of different pathways for that. And, looking at these results through and looking forward to the new stuff coming through our AgriScience cluster with this project, I'm optimistic to see it in the field trials.
Mm-hmm. Now, Peter wants to know, and so I'll ask the two of you and your experience with RNAi. Peter wants to understand, he observes that RNAi control seems to fail more rapidly than synthetic pesticide or the pesticides we have available right now. Peter, can you explain what you mean by fail? But Chris, do you have thoughts on that as far as what the experience has been as the effectiveness of this tool and the longevity, I suppose, of these different tools?
Yeah, I kind of briefly alluded to it a bit earlier on in terms of that lagging effect, right? Because it needs to kind of process through the organism, and so it's kind of like when we think about genes and we go from the DNA to the RNA, the expression of the RNA or how many I want to say copies, but amount of that RNA is going to dictate what the protein is going to look like in kind of a short form for that. RNAi as that deregulation isn't going to be perfect. And also, when we're thinking about RNAi, things can develop resistance, and there's multiple ways to have that resistance. For insects, they have ways to process RNA through their gut, right? For funguses, if we're trying to spray it on a plant, it's got to work itself to the pathogen as well. And then you still need that time to actually process and cause those effects. So those are some drawbacks. But what I am looking forward to is that rather than sometimes being a substitution, it's going to be complementary to some of our other practices. And I think the good example of this that we've already seen from Bayer in the VT4 Pro is that it's an additional mode of action to, say, the BT trait and such, right? Say if we're going to be using this for flea beetles, maybe we're going to spray, I have pyrethroid insecticide alongside with this. So we get a good contact, more instant kill, for lack of a better term. And then maybe we have some residual other impacts, so that way those flea beetle pressure kind of stays minimal.
And so if we do think about it as another mode of action, which it is, I mean, it potentially does become a novel mode of action in instances where we're already up against, some resistance as well. So it's a good way to sort of think about it. Let's go to, because I think this is a really great place to sort of land on, Chris's slide 5, if we could. And then Liping, Peter wants to know, in talking about your work, was CRISPR used to insert the cornstarch branching gene, or so it's my understanding CRISPR was used sort of after, not to insert the gene. But can you explain how CRISPR was used in that cornstarch model?
Yes, I should clarify. So this project involved two different technology. It is a combination of CRISPR technology and a traditional transgenic. Because, technical speaking, I don't think it's possible to replace directly with the cornstarch because canola has 6 SBE genes, and also cornstarch branching enzyme is so big. I doubt.
Okay, there are limits to the biology. Let's put it that way. We can't make everything mix. Okay, so this is a great slide, though, Chris, that we'll maybe sort of land on, and Liping, but maybe I'll start with you first. Obviously, there is great potential for CRISPR, for RNAi technology, but as we've outlined, I mean, there will be hiccups. There are hiccups. This is still a technology that we're bringing to market and learning more about. But this, I think, it does show where some of the challenges are. Liping, in your work, where would you say, what are some of the major hurdles that you're trying to overcome in the work that you're doing with this technology?
Of course, it is amazing technology. Yes, a limitation there. There is a limitation. I think the biggest one is the polyploid crop. So, like canola is tetraploid and wheat is 6 genome. So it has much more isoforms. Sometimes different cultivar, they have a modification. Like for example, in my project, I found that because canola is supposed to have 4 copies of a starch branching enzyme, but eventually I found there are 6. It is because there are 2 genes has duplication. I believe in other crop and other genes there will be frequent rearrangement, recombination, such kind of events. Yeah, that will be the major concern or bottleneck.
Mm-hmm. Plants are complicated. That's
Yes.
that's what we've come to. They're really
Yeah.
complicated.
It is
Yeah,
different from a model plant.
that's right. Wouldn't it be lovely if all of them were as simple as the model? But they are not. And it is one of the things that. So what I find fascinating about that comment, Liping, is that some of the hurdles to where traditional plant breeding, where they knock up against exactly that yield drag, or having to select and it takes a long whatever. I mean, some of these challenges still exist. It's just we're getting better at potentially streamlining the process, but these giant complicated genomes still cause a lot of challenges for sure. Chris, for you, in looking at something like this, and this is a great way to sort of think about where some of the challenges are. Where do you see bumping up against some of these walls? Is it the price point? Is it getting it commercialized? Is it the science part? Where do you see some of the hurdles?
I think the price is going to be one thing that's going to be maybe in the short term one of those barriers, but we can overcome that. The next thing is going to be like what is going to be the future of, say, this technology for this. We know that we're running out of, say, other modes of action, and we're getting resistance. And I think Peter and I are going to have a chat later on because there's so many good questions I'm itching to talk about. But uh, in terms of the resistance, we're thinking like I really love the examples for weeds, right? We have some weeds that can gain resistance by just producing many more copies, and it just overwhelms that herbicide. Or we may have a single site mutation where it's once it's the site is changed, it's complete resistance, that kind of thing. And I see just a quick thing on the chart: SIGs versus Higgs.
Yeah.
SIGs is spray
Sigs and Higgs, yes.
silencing, so that's the foliar. Higgs is
Okay,
host-induced
foliar.
gene silencing. Yeah,
Okay.
so that's like transgenic.
Okay.
But
Yeah.
for
Okay.
RNAi, at least, there's just because we may be starting to see resistance, and we know we found this, the Colorado potato beetle. There's been studies that you can develop resistance by forcing a bunch of it evolutionary. It's going to happen, right? But we do have some options, changing the structures of the RNAs, or potentially, finding ways to bypass it. There's options to use clay nanoparticles to coat and encapsulate the RNA for this. I talked earlier about using yeast as a platform for this. So just because we may see some of these resistance happening on the bench in a lab doesn't mean that we may have solutions later in the future. And I think it's good to keep having these discussions because we want something that's going to last, be effective, and be economical.
Mm-hmm. And I mean, to that point, that is the goal, but at the same time, realistically, we know that nature always does adapt as well. So we can't let the fact that pests will develop resistance to it be the barrier to adoption. I mean, that's like saying you don't make your bed because you're just going to mess it again. No, a made bed is much better to get into. So make your bed, everybody. Stop. Anyway, so but right, I mean, we have to be cognizant, I think, very cognizant of the resistance issue, and I think as an industry, we live and breathe this now of whether it's from the weed side or the fungicide or the disease side, but it also it's the stewardship of these different products and or traits becomes a key part of the conversation when we bring some of this forward and the commercialization of it. So that's a really good a really good discussion point, but definitely one of those ones that we are going to have to manage and move forward. So yes, Ray says it seems that the answer may be in your genes and how they are or get worn. How you wear your genes. GENES. Ray, that is brilliant. You win a gold star because it's a pun, and I love puns. So there you go. Okay, we're going to have to leave it there. We are out of time. Chris, you have many things I'm sure you want to talk to Pete about. I have many things that I would love to ask the two of you, but we are out of time. But I have learned a lot, and I appreciate it. I think I understand both of these, CRISPR and RNAi, better. But I also feel, like I often do with these topics, that I have learned just enough that I know nothing. So I have a lot more to learn. So that's the important part. So Liping, thank you so much for joining us. Your first time here on The Agronomists. I really appreciate it. Thank you.
Thank you. Really enjoy.
Yes. Good. And Chris, thank you so much for your enthusiastic response when I asked to do this episode. I really do appreciate it. And thank you so much for your time tonight.
Thanks. Always a pleasure.
All right. Okay. And everyone else, of course, next week is the September long weekend. So if you don't have your back to school stuff, go get it. So we won't have a show next Monday, but we'll return, of course, the Monday after that, 8 p.m. Eastern, as always. Thank you so much, everyone, in the comments. Great questions. I always appreciate it. And we'll be back in a few weeks. Cheers, everybody.